So, a very good morning to one and all. So, this is one of the very important and, of course, a very difficult topic to understand, that is amelogenesis. That is the process in which enamel matrix formation starts. So, basically, you divide it so that it will be easy for you to understand. So, broadly, amelogenesis, it takes place in two parts. That is, first there is formation of this organic matrix, and in the second part this organic matrix undergoes mineralization or maturation. So first we will see how this organic matrix formation takes place. So as we had also seen in the life cycle of ameloblast, the ameloblast, that is the enamel forming cells, begin their secretory activity only when there is a small amount of dentine, isn't it? So unless and until dentine is laid down, enamel formation will not begin. So the first layer of dentine is very important or critical. Okay. So once this first layer of dentine is laid down, the ameloblast lose the projection that had penetrated the basal lamina, separating them from the predentine. So why this happens? Because now the islands of enamel matrix that is produced are deposited along the predentine layer. And as enamel deposition proceeds, a thin continuous layer of enamel is formed along this dentine, and this is termed as the dentino-enamel membrane. So this is the first part that happens in amelogenesis. First, there has to be a layer of dentine, then there is the projections that were there, they disappear, so that the enamel matrix is laid down directly onto the layer of dentine, and this is your future dentino enamel membrane. Now second part that happens in the process of enamel matrix formation is there is development of this Tomes process. So the surface of ameloblast facing the developing enamel are not smooth. Okay, there is interdigitation of cells and enamel rods that they produce. So why they are not smooth? Because there has to be interdigitation, firm locking between the enamel and the dentine. Okay, and this is also because of one of the fact that the long axis of ameloblast are not parallel to the long axis of the rods. This is the second reason. So the projection of ameloblast into the enamel matrix, this is termed as Tomes process. So this you should know. What is Tomes process? Tomes granular layer is different thing. Okay, don't confuse between the two. That is Tomes process. It is the projection of ameloblast into the enamel matrix that they produce. This is Tomes process. Now this Tomes process, they also contain secretion granules as well as rough endoplasmic reticulum and mitochondria. Now there is one explanation between the rods and the ameloblast, enamel rod and the roughly hexagonal ameloblast. So it is said that at least two ameloblasts are involved in the synthesis of each enamel rod. And one interpretation of this keyhole shaped enamel rod. Now, as we have learned earlier in this chapter, the enamel rod is keyhole shaped. Okay, it is having a head and a tail, and the ameloblast is roughly hexagonal. Okay, so the bulk of the head of the rod is formed by one ameloblast, whereas three other contribute to the tail of the rod. So by this theory, four ameloblasts are involved in the synthesis of one enamel rod. One ameloblast contributes to the head of the enamel rod, and three ameloblasts, they contribute to the tail of the enamel rod. So in this diagram you can very well understand. So this keyhole shape, this is the head, and this is the tail of the enamel rod, and this roughly hexagonal is the ameloblast. So you can see one ameloblast is contributing to the head and three ameloblasts are contributing to the tail. So 1, 2, 3, 4. 4 ameloblasts are involved in the synthesis of one enamel rod. So I hope it is simpler by this diagram. The next part that happens in the formation of enamel matrix is development of distal terminal bars. So at the time Tomb's process begins to form terminal bar, they appear at the end of ameloblast, separating Tomb's process from cell proper. And what are these terminal bars? They are actually localized condensation of the cytoplasmic substances, and they are observed only during enamel producing stage of the ameloblast, but exact function is not known. Maybe they help in the firm interlocking between the enamel and the dentine, okay, but exact function is not known. So till now we have seen three parts. First, formation of enamel matrix, then second, development of Stone's process, and third, development of distal terminal bars. Okay. Now fourth thing that happens in the formation of enamel matrix is how is the shape or how are the ameloblast covering maturing enamel. So at light microscopic level, the ameloblast over maturing enamel are considerably shorter. As we had seen in the life cycle of ameloblast, the ameloblast, they are short columnar in the early stage. Then during the actual formation of enamel matrix, they become tall columnar. And then after the actual formation, when there is maturation occurs, again they become short columnar. So you should remember these three stages of ameloblast. So at light microscope level, the ameloblast over maturing enamel are considerably shorter than the ameloblast over incompletely formed enamel. So where enamel formation is going on, they will be tall columnar in shape. Now they have villous surface near the surface of enamel, and the ends are packed with mitochondria. So all these things, what they indicate, that this ameloblast is an absorptive type of cells, and ameloblasts are also transporting organic components from the matrix. So this also tells us that ameloblast is an absorptive type of cell. Okay. So the what is lost? Organic component and waters are lost in mineralization, and over 90% of initially secreted proteins is lost during maturation, because ameloblasts are transporting this. Okay, it is an absorptive type of cell. So 90% of initially secreted protein is lost during organic matrix formation, and there is a high content of this organic matter in prism sheath area, where the abrupt change in the crystal orientation occurs. And in an electron microscope, you can see several substages in transition of ameloblast from the formative to the maturative stage. We have seen this in the life cycle of ameloblast. And there are also shifts apparent associated with synthesis, secretion and absorption of this organic matrix. So, start. So this was the first portion of amelogenesis, that is organic matrix formation. So we have seen in that first there was what changes occur in the ameloblast during matrix production. Then we had seen what is Tomb's process. Then we had seen that there are terminal bars which are developing on the distal portion. Then we have seen what is the shape of the ameloblast. Okay, and how it is a type of absorptive cell, and how it is transporting organic matrix, that is 90% of mineral is lost during this formation phase. Now we come to the second part of amelogenesis, that is mineralization and maturation of the enamel matrix. So this also, it takes place in two stages. In the first stage, there is immediate partial mineralization in matrix segment, and interprismatic substances are laid down, and the initial influx of mineral content is almost 25 to 30%, and the first mineral that is laid down is in the form of crystalline apatite, that is hydroxyapatite crystals. Okay, so immediate partial mineralization, and in the second stage there is gradual completion of this mineralization. And this process of mineralization, it starts from the height, that is from the cusp or the occlusal areas, and then it proceeds cervically. Okay, and however at each level maturation seems to begin at the dentinal end, means they seem to end at the inside portion, lower portion. Okay, so simultaneously each rod matures from the depth, that is from in to the out surface. Okay, and the sequence of maturing rod is from cusps or incisal edge towards the cervical line. Okay, so from the outer portion it begins from the cuspal or the incisal area and then proceeds cervically maturation, while from the depth it from the innermost layer to the outer surface, that is at the dentinal portion, and then outwards towards the enamel. So that is the way how maturation occurs. So the maturation begins before the matrix has reached its full thickness. Thus it is going on in the inner first formed matrix and at the same time outer recently formed matrix. Okay, and the sequence of maturing rods is from the cuspal or incisal edge towards the cervical line. And maturation begins before the matrix has reached its full thickness. It is not like that all the matrix is laid down and that then it undergoes maturation. No, जैसे जैसे फर्स्ट लेयर ले डाउन हुआ है, सो उसका मैचुरेशन हो गया, एंड एट द सेम टाइम अदर layer of matrix is laid down, as we have seen this in earlier portion of this chapter, that enamel matrix formation is in layer by layer. Okay, एक ही पूरा bulk में matrix formation नहीं होता है. It is simultaneously going on in the inner and the outer portion. So that जैसे if the first layer of enamel matrix is formed in the innermost layer, okay, so that will be undergoing maturation, as well as the matrix which is laid down at the outer, that will undergo later. Are you getting me? So inner formed matrix will also undergo maturation, and simultaneously the outer formed matrix will also undergo maturation. So following this basic pattern, maturation begins before the matrix has reached its full thickness. Thus it is going on in the inner first form matrix and at the same time outer recently formed. So that is all about amelogenesis. I know it is very difficult to understand because most of the things, they are occurring in the electron microscopic level, and to understand this is difficult, but as I have divided into parts, like amelogenesis broadly it occurs in two parts, that is organic matrix formation and then maturation. And organic matrix formation also, there are certain steps, like how the ameloblast appears of the ameloblast of actual when it actually forms the enamel matrix, then what is Stone's process. If you remember this stepwise, it will be easy for you. Similarly, after finishing of matrix formation, there is mineralization or maturation. So this also, it takes place in two parts, that is immediate partial mineralization, and in the second stage there is gradual mineralization. And gradual mineralization also, it is taking place from the occlusal and incisal area, then it grows cervically, and inside first the innermost layer is laid down and then the outermost. So if you remember this, and naturally जहाँ पे पहले enamel matrix form हो रहा है, that portion will mature first, है ना? So if you remember this way, it will be easy for you. So I hope I have simplified and made it more clear. So that is all about amelogenesis. Thank you.